Okay, so in this video we're going to be talking about how cells will harvest energy from food. Here's an excerpt from your study guide. Here's a primer on cellular respiration. I think this is actually posted on Canvas already. There should be a video of the Amoeba sisters. Alright, so let's talk a little bit about the terminology to begin with. So your breathing or respiration is not the same as cellular respiration. So breathing, that's just your pulmonary respiration. You breathe air into your lungs, you let air leave your lungs. And so this refers to the gas exchange, oxygen in, CO2 out. Now cellular respiration, on the other hand, this is a process by which your cells, or more specifically your mitochondria, well actually I shouldn't say entirely your mitochondria, your cells will harvest energy from food molecules. So in order to get the full aerobic cellular respiration, you will need mitochondria, and your mitochondria need oxygen for it. So you do need respiration or pulmonary respiration in order to get cellular respiration, but the two are not the same. Okay, so for the most part, I would say that the majority of the energies that we consume on average is going to be coming from sugars. And so for that reason, we will be focusing on the breakdown of sugar for energy. So many molecules in food are converted into usable energy, or ATP, as we learned in the last chapter. That is the energy currency for our cells. And the sugar, glucose, so C6H12O6, remember this formula: 6 carbons, 12 hydrogens, 6 oxygens. That's the formula for most sugars. So the sugar glucose is the most common molecule used for energy. This is what it looks like. You don't have to memorize this chemical formula, this chemical formula, but you do need to know this equation here. So C6H12O6 is your sugar, your glucose, plus 6O2, 6 oxygen, dimolecular oxygen, yields 6 CO2 plus 6 water, H2O, and then plus energy in the form of ATP. So when you undergo full aerobic cellular respiration, this is the equation that you will be seeing. Sugar in, oxygen in, CO2 out, water out, energy out. Okay, so when it comes to cellular respiration, breaking down glucose, the first thing that happens is you break down that glucose. You break down the glucose into smaller pieces. So glucose is processed through a process called glycolysis. So glyco, you can associate that G with glucose. Glycolysis, so cutting glucose. What you're going to do in this process is you're cutting glucose into 2 smaller molecules called pyruvate. So there's 3 main metabolic processes that are used in the breakdown of glucose. Glycolysis is first. So as soon as glucose enters the cell, you will undergo the process of glycolysis to break it down. This process does not require oxygen, and it's going to be happening within your cytoplasm. Fermentation, that's kind of like your secondary metabolic process used to break down glucose, but fermentation doesn't fully break down glucose through aerobic respiration. This is actually an anaerobic process, and we'll talk about a little bit more about fermentation later. But to start off with, we'll go from glycolysis to the citric acid cycle, or the Krebs cycle. So glycolysis is when sugar enters your cell, you break it down into pyruvate. That pyruvate, in the presence of oxygen, will be sent to your mitochondria, and then within your mitochondria, you get this other preparation step called pyruvate oxidation, which prepares that pyruvate for Krebs cycle, and then you get Krebs cycle. So you've probably heard Krebs cycle maybe in your high school biology classes before. And so what you're doing here in Krebs cycle is you're taking acetyl-CoA, which we'll talk in detail about later, into its base components, or I wouldn't say base components, but you're fully breaking down all of those carbons in the all those carbon-carbon bonds that you find in sugar, and you're yielding a bunch of CO2. So those carbons will leave individually in the form of CO2. Now what happens inside of your mitochondria is going to require oxygen. So we call this aerobic. As soon as sugar enters your cell, you break it down through the process of glycolysis. This does not require oxygen. So we call that anaerobic. And then fermentation, which we'll get to towards the end probably, that is another anaerobic process. But as soon as you start sending stuff to your mitochondria, that's when you start using oxygen for things. And then lastly, after your citric acid cycle, through the process of your citric acid cycle, you've broken down all of those carbon-carbon bonds. What you do is you generate this proton gradient, or you send those electrons. So you're actually harvesting electrons. You send those electrons to the electron transport chain, and then your electron transport chain will then conduct the processes needed to create ATP. So that's your final step in full aerobic respiration. So this is kind of how it would look like visually. Glucose enters the cell. It's broken down through a process called glycolysis. Now you can ferment it, but we'll talk about that at the end, when there's no oxygen present. However, if there's oxygen present, what you're going to do is you're going to send this glucose, which turned into pyruvate, you're going to send this pyruvate to the mitochondria. Within your mitochondria, you're going to oxidize that pyruvate to break it down into acetyl-CoA, and then within still your mitochondria, it undergoes the citric acid cycle. And so here, you're plucking away carbons in the form of CO2, same with pyruvate oxidation, you're plucking away carbons in the form of CO2, and you're making some ATP in the process. But the main important part here, so we could start with glycolysis. The main part of the show here is this NADH here. So these are molecules that carry electrons. What you're doing is you're stripping away glucose of its electrons. In the process, also breaking bonds. You're harvesting a little bit of ATP, but really that's nothing compared to what you get at the end, your payout. So through the process of this full oxidative respiration, you are harvesting these electrons, and they are carried by electron carriers. So NADH is considered to be an electron carrier. Same with here, pyruvate oxidation, you're harvesting electrons carried by electron carriers. Citric acid cycle, look, 6 NADH, so 6 electrons that you'd be carrying through this electron carrier. And then FADH2 is kind of a less potent electron carrier, but still an electron carrier nonetheless. And so what's creating the bulk of your energy is actually when we actually utilize these electrons and we plug it into the respiratory chain, so the electron transport chain. And so just as review, the formula, you've got C6H12O6 plus 6O2, which makes 6 CO2 plus water, 6 H2O, plus around 36 ATP. Oxygen's being put in, and you're pumping water out within your respiratory chain. Okay, so electron carriers. So we just kind of talked about electron carriers. NADH, that's going to be carrying an electron. FADH2 is also carrying an electron. However, if you see something like NAD+, it is not carrying an electron. FAD, not carrying an electron. So these are the guys that are going to pick up the electrons kind of in the form of a hydrogen. Hydrogens carry electrons, and so you bring that electron aboard, or you bring that hydrogen aboard containing that electron, and now you're able to utilize that electron. And so this is just showing an enzyme. This represents an enzyme actually attaching that hydrogen plus electron to an NAD plus to make NADH. Okay, so now we're going to kind of deep dive into the first step, glycolysis. So this happens in the cytoplasm. So glycolysis is going to happen in the cytoplasm, and what's going to happen here is we actually invest a little bit of energy to get more energy out. So we're actually using 2 ATP to break down glucose. One glucose splits to make 2 pyruvate, and 4 ATP is what's yielded. So we actually put 2 ATP in, and we get 4 ATP out, making the net gain of ATP 2 ATP. We're also getting some NADH, which we use much later in the electron transport chain, or for the electron transport chain. So here's where we invest the ATP. So we have C6, carbon 6 carbons. You add a phosphate, now you got 2 phosphates on this 6 carbon sugar. We split it in half. Each of these 3 carbon sugars have phosphates. And what happens is you harvest electrons. So you bring NAD plus in, you swoop away that electron and that hydrogen from the molecule, and as a result you get to NADH. Then this molecule picks up an inorganic phosphate, so not from ATP, but from phosphate floating around in your cytoplasm, picks up a phosphate. And then what we do is we grab that phosphate by using ADP to combine with that inorganic phosphate to make ATP. So we call this substrate level phosphorylation. So we make some ATP. So now we've broken even. We put 2 ATP in, we've gotten 2 ATP out. Now, atoms are shuffled around within the molecule. So atoms are shuffled around. We don't have to know all of the details of what atoms move where. But this is all happening through a multi-step process conducted or orchestrated by enzymes. So enzymes are going to be shuffling around atoms, and then you make 2 more ATP. So at this point, you've netted 2 ATP. And then what you're left with are these 2 3-carbon pyruvate molecules. So here we go. We have phosphorylation, so your investment phase. Then you cut this 6-carbon sugar in half, lysis, the lysis phase. And then we have our dephosphorylation. So we're taking that phosphate back, taking the substrate or that inorganic phosphate that we attached to it, as well as the phosphate that we put onto it to begin with. So dephosphorylation step. So as a result of glycolysis, what's the net gain of ATP? What do you think? So the answer is 2. We put 2 in, we get 4 out. So the net gain will be 2. Okay, fermentation, we'll go back to that later. So we're assuming that we're in the presence of oxygen. So the cell is in aerobic conditions, so we're not starved of oxygen. And so what we do is we send that pyruvate to the mitochondria. So within the mitochondrial matrix of eukaryotes, this is where you're going to get aerobic respiration. So aerobic just means with oxygen, and this is a 2-step process. So before we can jump right into Krebs cycle, we have a preparation stage. We call this pyruvate oxidation. This is going to happen in the matrix of your mitochondria. So glycolysis happened out here. We create that pyruvate. Pyruvate diffuses through the bilayer of, or there's actually two layers of membrane in the mitochondria, and it lands in the matrix. And so then pyruvate oxidation begins. So we have this 3-carbon sugar or 3-carbon molecule called pyruvate. And what happens is this pyruvate, so this 3-carbon molecule, loses a carbon as CO2. So CO2 is plucked away and is now called an acetyl. So acetyl groups are generally 2-carbon molecules, so we just get an acetyl group. So acetyl combines with coenzyme A to make acetyl-CoA. So here's your pyruvate, and you undergo pyruvate oxidation. You have this 2-carbon acetyl group, and you've added this coenzyme group to it. So that carbon leaves in the form of CO2, and then in the process of all of this, we are harvesting electrons in the form of NADH. So NADH is produced per pyruvate molecule. I probably meant to put 1. Or, yeah. Okay. All right, so now that we've created acetyl-CoA, now it's ready for the citric acid cycle, or the Krebs cycle. So citric acid cycle is going to happen in the matrix. Acetyl-CoA binds to the starting molecule to form citric acid. So originally, or floating around in the matrix, you have all of these molecules actually. But what happens is that acetyl-CoA is bound to an enzyme, and that enzyme also brings in oxaloacetate, which is a 4-carbon molecule. So you got this 2-carbon molecule, which is now fused with this 4-carbon molecule to make a 6-carbon molecule we call citrate. So that's where citric acid cycle gets its name. The first product that we get in this cycle is citrate. Okay, so, and we don't have to know all of these other names here. It'd probably be beneficial for you guys to know citrate, probably oxaloacetate as well. Same with acetyl-CoA, and same with pyruvate. But isocitrate, alpha-ketoglutarate, we don't need to know this stuff here. So what happens is you combine oxaloacetate to acetyl-CoA to make citrate. Citrate is, the atoms within citrate are shuffled around through an enzyme. We make isocitrate, so iso probably implying isomer. It's an isomer of citrate. And then we continue with this citric acid cycle. Cycle because what we start off with is actually what we end with. And so you shuffle around atoms, you pluck off some CO2, you harvest some electrons, you get some NADH, you pluck off some more CO2, or you pluck off some more carbons in the form of CO2 by breaking those carbon-carbon bonds. You harvest more electrons in the form of NADH, and then eventually you make a little bit of ATP. So it's not a significant amount, but it is still a net gain. So you do harvest some ATP, and then you harvest a few more electrons in the form of FADH2. You add some water at some point. You harvest a few more electrons through NADH, and then you're left with oxaloacetate, which now binds to the enzyme containing acetyl-CoA from the outside, and you just continue the cycle. All right, so let's look at this part here. So acetyl-CoA binds the starting molecule to form citric acid or citrate. Those can be used interchangeably. Making NADH and ATP molecules shuffle around and CO2 is released. So out of all of the processes involved in cellular respiration, your citric acid cycle creates the most CO2. Making NADH and FADH2, the starting molecule oxaloacetate is regenerated. Okay, so the enzymes of the citric acid cycle are located where? Enzymes of the citric acid cycle. Think about where the citric acid cycle is occurring, in the matrix of the mitochondria. So it makes sense that floating in that matrix are probably enzymes, and maybe you have some enzymes embedded in the inner mitochondrial membrane as well. So mitochondrial matrix and the inner mitochondrial membrane would be the correct answer. Outer mitochondrial membrane wouldn't make sense because that's happening maybe outside of the mitochondria, or if it's embedded in the outer mitochondrial membrane, maybe within its intermitochondrial space, which we'll look at later. Not in the nucleus, not in the cytoplasm. Okay, so the final step of aerobic cellular respiration is the electron transport chain. So all of those NADH and all of those FADH2 are brought to this part of the mitochondria. One thing that I should probably talk about is that the mitochondria actually contains two membranes. So you have this outer membrane, but you also have this inner membrane. Remember, when we did look at the mitochondria, we saw how you had the cristae of surrounding the matrix. So the cristae are those folds in that inner membrane. That's because there's actually two membranes within the mitochondria. So here's your outer membrane, here's your inner membrane. So anyways, from outside you bring all of this NADH, and then from the matrix you bring all this NADH to these electron transport proteins. And so what happens here is NADH drops off an electron, yielding H+. So H+, what that implies is that it's missing something negative. What is it missing? It's missing its electron. So that electron was taken away from that hydrogen atom. Now we generally associate anything with high H+ concentration to be acidic, and so therefore making, essentially, an acidic environment. So NADH strips away, or NADH drops off its electron and yields that hydrogen, making NAD plus as a result. So now NAD plus is sitting around in the matrix. And what this protein does from this potential energy, so NADH is a type of potential energy. It's kind of like this intermediate currency. NADH and FADH2 are these intermediate currencies. What we do is we convert this potential energy into a kinetic energy of moving a hydrogen against its concentration gradient into this intermembrane space. So through this, or actually I'll talk about what we actually do with these hydrogens later. So you drop off your electron, you convert that potential energy into the kinetic energy of moving this hydrogen into this intermembrane space. Then that electron moves from protein to protein, so from protein to protein to protein to protein. And in the process, you're converting that kinetic energy into another kinetic energy. So you're kind of like, you're churning. What you're doing is you're kind of, churning the wheel of a wind turbine, for example. You're moving hydrogens into the space. So electron moves, pump hydrogen, moves, pump hydrogen. And so now you're pumping all of this hydrogen into this intermembrane space. Eventually that electron needs to stabilize itself, and so what happens is it actually binds to oxygen, and that oxygen just picks up a hydrogen within the matrix, and you form water. So no biggie. You just form water. You don't have those free electrons floating around damaging your cells. But what you're left with is a high density of these hydrogens in this intermembrane space. And so what we do, so first off, these hydrogens, you have a lot of them in this intermembrane space. You have very small amounts in the matrix of the mitochondria. And so these hydrogens really, really want to move into the matrix of the mitochondria. And we do this, we allow this to happen. So we'll let this hydrogen move through, but we let it move through this enzyme called ATP synthase. So as the name implies, you're probably going to be synthesizing ATP. And so this is kind of like a dam where we use the movement of water to create energy. So you have all of this potential energy, all of these hydrogen ions that want to move through. We let it happen, and what it does is it churns this enzyme to form ATP from ADP. So you've got an ADP sitting on this enzyme. The movement of these hydrogen ions moving through here allows this enzyme to form ATP. So it's kind of cool. We started off with glucose, which is a type of potential energy. We converted that potential energy into NADH and FADH2, or the electrons, which are potential energy. We converted that potential energy into this potential energy, this hydrogen gradient in this intermembrane space, and then we converted this potential energy into the potential energy we can finally use as currency within the cell. That's so cool. Okay. So this is, I have this in place to kind of describe, to use an analogy to describe that those hydrogens moving and why they would move. So we understand diffusion already. So, you drop some dye into some water, and the natural tendency of these dye molecules is to move to where there is less of itself. It moves towards its concentration gradient, right? And so eventually you reach equilibrium when you have an even distribution of those dye molecules. So if you're looking at a battery, for example, usually you have a positive side and a negative side. In your negative side, it's negative because you have a lot of negatively charged electrons. And the way we make batteries do stuff, or the way we harvest the energy contained within your batteries is, well, first off, you have to think of the potential energy here. You've got a ton of electrons in this negative side that really want to go to this side that's absent of electrons entirely, so the positive side. So tons of electrons in here no electrons in here. However, there's a barrier in between them, so they can't reach equilibrium. So what we do is, let's say we grab a wire and we bring it along, so a wire that electrons can travel through, and in the process of bringing that wire to the positive end where the electrons want to go, we run it past a light bulb. And when we do that, these electrons surge through that wire, and in the process, well, now your light bulb lights up. And what happens is they get to go where they want to go, towards this area absent or devoid of electrons. So we kind of do that in our mitochondria, where these hydrogens really want to be down here, and in the process, we churn it through ATP synthase to make ATP. So instead of lighting a light bulb, we just utilize that energy to make a different type of potential energy. So it uses the hydrogen gradient to synthesize ATP. And that whole process, the process of moving this hydrogen through ATP synthase to generate ATP, is called chemiosmosis. The process of hydrogen coming back across through the ATP synthase along its concentration gradient to form ATP is called chemiosmosis. So during chemiosmosis, let's see what's true. So energy is released as hydrogen ions move freely across the mitochondrial membrane. They don't. Your hydrogen ions don't freely move across the mitochondrial membrane. Remember, they're moving through ATP synthase. So obviously they're inhibited in some way. ATP is synthesized when hydrogen ions move through a channel in ATP synthase. That's true. Let's look at the other stuff. A concentration gradient is generated when large numbers of hydrogen ions are passively transported from the matrix of the mitochondrion to the mitochondrion's intermembrane space. No. Your hydrogen ions are not they're not passively transported from the matrix to the intermembrane space. They are actively transported. Remember, you're using that release of electron, that movement of that electron, as fodder for moving the hydrogen into that intermembrane space. So it's a type of active transport. We're not using ATP per se to do it, but we are using a form of kinetic energy. And then lastly, we have hydrogen ions serve as the final electron acceptor. The final electron acceptor is oxygen. The final product is water, in this case here. And that's actually something very important to remember. The final electron acceptor in the electron transport chain, the final electron acceptor in the electron transport chain is oxygen. Okay, so now we've talked about creating ATP through ATP synthase, through the electron transport chain, through Krebs cycle, through pyruvate oxidation in the presence of oxygen, so aerobic respiration. However, what if our cell is starved of oxygen? What if we've exerted our cells so much that they've depleted their oxygen? Well, they still need ATP, and so out of desperation, what happens is something called fermentation. So we're going to move back up to fermentation here. So in fermentation, we're converting pyruvate into organic molecules. Does not require oxygen. And so as a result of fermentation, what we do is we get, in us mammals, we're going to synthesize lactate, which is still a very large organic molecule compared to CO2, for example. CO2, that's, this teeny little molecule that is, it just dissipates. It's tiny, and there's not much potential energy in it. However, lactate still has a lot of potential energy. So when we undergo fermentation, we actually waste a lot of potential energy, waste a lot of materials. Okay, so fermentation. This is going to happen in the cytoplasm. It is anaerobic, so without oxygen. And what happens is pyruvate is converted into lactate in animals. And if we're dealing with yeast, alcohol plus CO2. So that's why when you're fermenting wine, for example, you put that balloon on top and you'll see that balloon expand. That's because CO2 is actually getting generated a little bit, but you're still left with alcohol. That's the active ingredient you're looking for. So in fermentation, what's happening is you're regenerating NAD+, and you're getting an incomplete breakdown of pyruvate, or an incomplete breakdown of glucose, if you want to think about it that way. Okay, so you've got glucose, which undergoes glycolysis. Now you get pyruvate. Instead of sending it to the matrix of the mitochondria, because you are starved of oxygen, you then determine, I'm going to ferment this in the cytoplasm to get ATP as fast as possible. Now, when we look here, one of the rate limiting factors of glycolysis is actually NAD+. If you have no NAD+, glycolysis stops. So NAD is required for it. Even though it's an electron carrier, we use this to send to the matrix of the mitochondria, it is still a rate limiting molecule. So if you have no NAD+, you no longer do glycolysis. Remember, the net yield of glycolysis is 2 ATP. Very small amount, but if you're desperate, it works. And so what happens in fermentation is you take, and this is even more wasteful, but you take NADH, so you have the fully loaded electron carrier, and you deplete it. You actually add those electrons to your pyruvate to make lactate. So you actually waste those electrons in order to get to NAD+. But what you do here is you use this 2 NAD+ to feed back into glycolysis, yielding you to net ATP. So fermentation actually doesn't make any energy on its own. What it does is it potentiates glycolysis to make 2 more ATP. So a lot bit less efficient than aerobic respiration. So fermentation coupled with glycolysis yields you to net ATP, whereas if you were going to undergo aerobic respiration, if you were to undergo aerobic respiration, you're getting a total of 32 to 38 ATP, depending on the conditions of the cell. So fermentation coupled with glycolysis has a net yield of 2 ATP with glycolysis. Okay. And so this is just the recap again. What I'm going to be doing in the next video is drawing it out for you guys to slow it down a little bit. Here's a summary of everything for you visual people. And then this is just a reminder that, we don't only use sugar for energy. Sugar is not the only thing we break down for energy. We know from polysaccharides, many sugars, we get sugars. And then from sugars, we throw that into the cytoplasm. Glycolysis happens. We get pyruvate, acetyl-CoA in the mitochondria, and then Krebs cycle within the mitochondria. However, we could break down other macromolecules, so like lipids and fats. What we do is we'll break down the fatty acids through a process called beta oxidation, create a bunch of acetyl-CoA, throw that into the matrix of the mitochondria, and let that churn out within Krebs cycle. Harvest those electron molecules electrons through the intermediate NADH and FADH2, and then plug that into the electron transport chain. We can also break down amino acids and nucleotides, amino acids through a process called deamination. And because amino acids are so variable, sometimes we can actually make acetyl-CoA with them, but we could also make intermediates within the Krebs cycle from the amino acids, from breaking down those amino acids. And in the Krebs cycle, we churn out some NADH and FADH2. So it's not only sugars that give us energy. And so this was just a review. I don't think I listed this one on Canvas.